So secondary, let's talk about secondary structure. So primary structure is held by peptide bond. Now secondary structure is held by hydrogen bond. And this hydrogen bond is a non-covalent bond. So many people confuse what is type of, it is basically attraction between two electronegative group inside the molecule. So this is represented by 3 dots. So hydrogen bond is a non-covalent bond. So secondary structure is stabilized by hydrogen bond. And these are secondary structure. You can see alpha helix and beta sheet are the main thing. Alpha helix and beta sheet. We can say beta pleated sheet. Anyway, alpha helix and beta sheet are secondary structure. Now, other secondary structure you will see in collagen. Collagen, you will see here, you will see alpha helix is right-handed. Right-handed. In collagen, you will see left-handed alpha helix. This left-handed alpha helix, and that also there will be triple helix. We will talk about in detail about collagen. There will be 3 protein together. There are 3 protein together. So this is triple helix. And this is called super secondary structure. That is why collagen is very strong. This is also sometimes called super secondary structure. Okay, so this is collagen. Collagen. And this collagen is also held by hydrogen bond, plays very important role in collagen also. We will talk in detail about collagen synthesis and processing collagen. Don't worry about it. First, let's focus on this alpha helix and beta pleated sheet. Alpha helix and beta sheet. So those both are held by this hydrogen bond. Hydrogen bond between carbonyl, and this is also tested. This is also tested in NBME and other exams. There is bond between this carbonyl free, you know, there is free carbonyl carbon here at the peptide bond and N group. So there is hydrogen bond between this and other amino acid residues. When amino acid is present into protein, it is called residue, amino acid residue. It is not a complete amino acid. So amino acid, so this will form bond with this H. So I will show you how it forms this bond between this oxygen of carbonyl oxygen and hydrogen of this amine group. How it forms like attraction, not actually bond. This bond is basically attraction, like electrostatic attraction. Attraction between negative charge and positive charge. So you can see alpha helix is the most common. This is the most common. Alpha helix is the most common and stable control, most stable, more stable than beta sheet. So alpha helix is the most common. And you know, and this we have already done hemoglobin and myoglobin. And there I have already told you this hemoglobin and myoglobin has all alpha helix. This is also a question. It has got all alpha helix. There is no beta sheet in hemoglobin and myoglobin. All alpha helix. This is also a question. So understand, remember this point. Understand this point. Hemoglobin, myoglobin, whole molecule has secondary structure of alpha helix. No beta sheet there. So this, understand this. And whereas it is virtually absent in, virtually means nearly absent. So this chymotrypsin enzyme that digests protein present in our intestine, secreted from pancreas, is almost all beta sheet. There is no alpha helix. So these are this is actually very important. You need to understand hemoglobin, myoglobin is all alpha helix. So alpha helix is right-handed, as I've already described. This is right-handed structure, and then polypeptide bond form backbone. NCC, you know, the polypeptide, the peptide bond forms the backbone, and side chain of the amino acid extend outward. You already know this. Now, structure is stabilized by hydrogen bond. How? I will tell you how this hydrogen bond stabilizes structure. I will tell you. Now, each turn is about less than 4 amino acid residue. How? That will also I explain you. Let me. And distance between each amino acid residue is 1.5 angstrom. I'll describe this point through the picture right now here. You can see here, this is amino acid. You can see, can you see this is like a sort of triangle? Now this amino acid, there will be a little bit of folding, but you can see this is you can see this is like alpha carbon. This is alpha carbon. Now this alpha carbon is folded like this. Okay, now this will fold like this. So it is very difficult to show here, but anyway, this will fold like this. Again, I will show like this. So this will fold like this, and then like this. Again, the helix will go like then. So that way. Now you can see here, there is alpha carbon. You know, alpha carbon is there, and then with this carbon, carbonyl carbon is there, and with this carbonyl carbon NH. This is peptide bond. Now alpha carbon, again carbonyl carbon, and then peptide bond, N, and then. So if you see this, so this will form bond with the fourth. In that sense, it will form this carbon. This, let's count from this. This is the N terminal. You can see this is the first amino acid, and this is first amino acid. This is carbonyl, CO. Now this CO, when it folds the fourth amino acid, it will make bond with this NH of this fourth amino acid. NH of this fourth amino acid. So first we'll make hydrogen bond with the fourth amino acid. Then you can see you add 3, then fifth will make the hydrogen bond with, add 3 to this, with 8th amino acid. 8th will make hydrogen bond with, add 3, 11th amino acid. The first will make hydrogen bond with the 4th amino acid. First CO will make the hydrogen bond with next 4th NH2 group. So that way it goes on. So you can see many hydrogen bond. Hydrogen bond is very important for stabilizing this structure. Now you can see that the distance between height between these two amino group is 1.5 angstrom or 0.15 nanometer, and the pitch is 5.4 angstrom. So you don't have to memorize this pitch and this. So just remember, one pitch has about nearly 4 amino acids. 3.6 amino. That's it. So you don't have to just, and remember this alpha helix and beta sheet is stabilized by secondary hydrogen bond. Can you see this is a carbon? Can you see carbon? This is oxygen. Okay, carbon, oxygen. Now you can see carbon, and this is nitrogen, and this is hydrogen. So this is electropositive, and this is electronegative, and there is attraction between this. This attraction is called hydrogen bond. This hydrogen bond is holding this prime. This hydrogen bond is helping the secondary structure to form this spiral, because there will be traction like this. So this attraction holds this structure. Did you understand now?
Yes, sir.
So secondary structure is held by hydrogen bond. Remember, this is a very important point. Secondary structure is held by hydrogen bond. This hydrogen bond holds secondary structure. That is very important. Now in the alpha helix, you can see in alpha helix, you know, in any protein, the initial NH is free, because I'll tell you. Can you see? Initial NH group is free, and terminal COO group will also be free. Other N and CO group will be in bond, and that is why this is a positive charge, and this is negatively charged. Negative charge. Now you can see any protein alpha helix. You can see one terminal of alpha helix will have free NH2 group, and that is actually bond NH3. This is positively charged, and terminal group will be. So all alpha helix will have terminal negative charge, and head will be positive. See, this NH3 is called head. As you see in genetics also, there is head, 5' head, and tail. So this carboxy is tail. Okay, so head is always positively charged and tail is negatively charged. Did you understand this?
Yes, sir.
Now let me talk a little bit more about this. So what happens, this alpha helix, if you see from the top, and this alpha helix inside this inside membrane can have this type of alpha helix. I'll tell you this type of alpha helix. So this type of alpha helix will form water channel inside protein, inside lipid. So it will form a water channel inside lipid. You know, this part of the alpha helix has to be hydrophobic because it has to interact with lipid. So some alpha helix have a half part hydrophobic phase and half is hydrophilic. This hydrophilic phase will allow movement of water from outside to inside of the cell. So this alpha helix can have this two phase. If you see from the top or the bottom, cross section of alpha helix can have like this. And this type of alpha helix is part of, not alpha or all alpha, this type of alpha helix is part of this water channel. For example, aquaporine. You will study in renal system. So this is a very important. Now you can see secondary structure of beta pleated sheet. Beta pleated sheet can be parallel or anti-parallel. So beta pleated sheet, see, you can see that beta pleated sheet can fold like this. So this is, let's say, NH3 group. So it will fold like this. This will fold like this. So what do you think? What type of this folding is? Parallel or anti-parallel?
Parallel.
Arrow going this side and this side. You can see here.
Anti-parallel.
Anti-parallel. This is anti-parallel. Okay. So whenever you see a protein, for example, you already see chymotrypsin, a protein had with only beta sheet. That protein will have always anti-parallel, and there will be hydrogen bond here. This hydrogen bond between carbonyl and NH2. So this will hold this. So this will hold this whole structure of beta cell. Now, so this is anti-parallel. So what will happen with parallel? So parallel, what happens is when the protein fold like this, and then there is alpha helix in between, and then again protein folds like this. So what do you see here? What do you see here? You can see. So this is the flow of this protein. This is the flow. So this alpha helix will be, this beta sheet will be held by parallel hydrogen bond, and this is parallel beta sheet. So parallel beta sheet always will be will have a combined alpha and beta. Parallel beta sheet is never a pure. Parallel is never a pure. Did you understand this?
Yes, sir.
So this will fold like this. So alpha helix, then so this will come like this. So simply you will say this is like this and this is like this. So this is parallel beta sheet. So you can see this type of folding and a sort of loop-like folding, and here it will be alpha helix. So this is parallel and anti. Parallel is impure. P for impure, and anti-parallel is pure. Understand this? So this is how beta sheet folds. So you can see this is a parallel polypeptide chain in beta pleated sheet. So distance between two adjacent amino acids, you can see, is like this, 3.5 angstrom. So stabilized, you can see there is hydrogen bond between CO and NH. NH and this is CO. So you can see hydrogen bond is holding this anti-parallel. This is anti-parallel beta sheet. Now parallel beta sheet also will be held by this. Little bit difference will be there. I have already told you how it holds, so you can understand. Beta sheets bends when bends will be formed in many protein, and this abrupt U-turn folding of chain. This is called U-turn, and this is called beta turns. We will talk about beta turns right now. So this is beta sheet. Just understand the beta sheet is the simple folding like sheet, and it is this sheet is held by hydrogen bond. And this is two types, parallel and anti-parallel. You can see the example of parallel. Parallel is fibrin, a spider fiber. So you can see spider fiber is basically protein. The net of web of a spider is basically protein, and that is anti-parallel, pure protein, pure beta sheet. This is pure beta sheet. And this flavodoxin is parallel, it is impure. And carbonic anhydrase has both parallel and anti-parallel. So these are the examples. And silk, you previously the clothes, silk clothes extracted from the silkworm, that silk also is a beta sheet. You can say this beta sheet, we designate this through this ribbon diagram. This is called ribbon diagram. You can see this is parallel, and now you can see this is parallel, 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 and here you can see anti-parallel. So this is both like this. Both will be like this. There will be certain part will have parallel, and then certain part will have anti-parallel. So parallel and anti-parallel can come together. This is beta turns, and beta turns, you see this beta turns are the turns that will be made by this beta sheet at the end of this beta sheet. So this is beta turn, and this beta turns have a very specific type of amino acid generally. The two types of very flexible amino acid is glycine and very inflexible. There are two amino acids. The most flexible amino acid is glycine and least flexible is proline. So this both amino acids are present here. Because of flexibility, it will give, glycine will give a freedom for forming a turn. Here you can see glycine, but proline lacks flexibility, and proline is a bit kinky amino acid, so it will provide kink. So kink is helpful for formation of this beta turn. So remember, these two amino, glycine and proline, are present at the beta turns. You can see proline here. Proline is a kinky type of amino acid. Just remember glycine and proline. And proline, glycine is very flexible. So glycine and proline, they are present in beta turns, also in beta sheet. But this same glycine and proline are not present in alpha helix. They are present in very least quantity in alpha helix. Why? This is a very important point. So you can see alpha helix has very less glycine and proline, very less, the least. But beta conformation, beta turns, you can see they have highest glycine and proline. Why does alpha helix not have this glycine and proline? Because so this glycine is very flexible. Again, the answer is same. Glycine is very flexible. That is why it will not actually be good to hold this alpha helix. And this is very rigid. Proline is very rigid. That is why it will not allow. It will not allow this folding. This proline will not allow folding, and this will be too flexible to adapt into this folding. That is why glycine and proline is very less in alpha helix. It is very high in beta turns and intermediate in this beta conformation. So in beta conformation also it is very less. You can see the list among all. But it is more than, you can see more glycine is more than that is present in alpha helix. But in both beta sheet and alpha helix, this proline and glycine is less because it is not very feasible to form this sheet and helix. But it is very feasible for formation of turns. Why is proline low or absent in alpha helix? Because of rigidity. Okay, rigidity. It's rigidity. And then if we talk about this glycine, because of its flexibility, one is very rigid, one is very flexible. So you can say this is glycine. You can remember this is proline. You can remember easily. So this amino acid is like P. This is like P. So this is amino, and this is amino. This is acid. Actually, the propane side chain will form bond with amino. This is a close. This is called amino acid. You can see amino group is attached with 2 carbon here. Amino acid. That is why this is secondary amino group. Only one amino acid which has secondary amino group. So we will talk in detail about amino acid. Then I will talk in detail about proline. Proline is, that is why this proline is rigid. This structure makes it rigid. Cyclic structure makes it rigid. It has got geometric constraint. And then nitrogen in peptide linkage does not contain H atom that is required to form hydrogen bond. So it also does not have hydrogen bond available for formation of hydrogen bond that is required during stabilization of helical structure. So it is not feasible for formation of hydrogen bonding also. So secondary structure of protein is stabilized by which of the following bond?
Hydrogen.
B.
Secondary structure of protein are alpha and beta structures and are stabilized by formation of hydrogen bond between first and fourth. Okay. So this is not NH. This is first CO of first to NH of fourth. CO of first to NH of fourth. CO to NH. CO NH. CO of first amino acid will form its successive fourth. First, fourth, first, fourth, first, fourth, second, fifth, third, sixth, that way.